Welcome back to Neutralizing COVID, the monoclonal antibody story, the podcast where we unpack the science, strategy, and real world impact of monoclonal antibodies in the fight against COVID 19. I'm your host, Dr. Shikha Agrawal, and today we are doubling down on defense as we explore the power of antibody combinations. This is Double Defense Lesson from Regeneron's REGEN-COV cocktail. Back with us is Dr. Aiswarya Chaudhuri, a seasoned researcher in nanomedicine and drug delivery systems for cancer treatment. Welcome back, Dr. Aiswarya.
Thank you, Dr. Shikha. It's good to be back on the podcast. I've been looking forward to this.
So, last time we discussed a single monoclonal antibody. REGEN-COV, however, is famously a cocktail of two antibodies. Could you explain to our listeners why researchers would choose to combine antibodies rather than just using one?
Certainly, Dr. Shikha. The primary rationale behind an antibody cocktail is to combat the virus's ability to mutate and escape the treatment. Viruses, especially the RNA virus like SARS-CoV-2, are constantly evolving. So if you use a single antibody targeting one specific spot on the virus, there is a risk that the virus could mutate that spot, making the antibody less effective or even ineffective. Now, by using a cocktail of two or more antibodies that bind to different, ideally non overlapping sites on the virus, you significantly reduce this risk. It's like trying to pick two locks simultaneously instead of just one. So the virus would need to develop multiple mutations in the right places at the right time to escape the effects of both antibodies, which is statistically much less likely, at least initially.
That makes a lot of sense. A sort of double layered defense. How were Casirivimab and Imdevimab, the two components of REGEN-COV, actually developed and chosen for this cocktail?
Regeneron has a well-established platform for rapidly discovering and developing antibodies. For REGEN-COV, they screen thousands of antibodies, both from humans who have recovered from the COVID-19 and from their proprietary mice that are genetically engineered to produce human antibodies. The two antibodies, that is the Casirivimab and Imdevimab, were selected because they were both highly potent neutralizers of SARS-CoV-2. Crucially, laboratory studies confirm that they bound to distinct non-overlapping regions on the spike protein receptor binding domain, that is the RBD. Now, the RBD is the part of the virus that latches in onto the human cells to initiate infection. Now, by targeting the two different sites on this critical domain, the cocktail aimed to provide a robust defense.
Fascinating point. They are both targeting the main entry key of the virus, but hitting different parts of that key. Can you elaborate a bit more on their precise mechanism of action once they are in the body?
Exactly, Dr. Srikant. Both Casirivimab and MDVimab are neutralizing antibodies. Their primary job is to bind to the spike protein of the SARS-CoV-2. By doing so, they typically obstruct the virus's ability to attach to the ACE2 receptor of human cells. Now, if the viruses can't attach to or enter the cells, it cannot replicate and cause the widespread infection. Because they bind to different sites on the RBD, even if a mutation occurs that slightly reduce the binding of one antibody, the other could still effectively neutralize the virus. These synergies the core idea between the cocktail strategy to maintain efficacy even as the virus attempts to evolve.
And how effective was Regen-CoV when it was first introduced, particularly against the earlier variants of SARS-CoV-2 that were circulating?
REGEN-COV showed a significant promise in clinical trials, especially for treating mild to moderate COVID-19 patients who were high risk of progressing to severe disease. Studies demonstrated that when given early after infection, the cocktail could reduce the viral load lessen the severity of the symptoms, and significantly decrease the risk of hospitalization and death. It received the emergency use authorization from the FDA in late 2020. During the period when the earlier variants like the Alpha, Beta, and Delta were dominant, REGEN-COV was a valuable tool in the therapeutic arsenal.
That's a powerful impact. But as we know, the virus didn't stand still. How did REGEN-COV cocktail fare as newer variants like Omicron emerged with many more mutations?
This is where the ongoing challenge of the viral evolution becomes starkly evident. The Omicron variant, which was emerged in late 2021, carried an unprecedented number of mutations in its spike protein, particularly within the receptor binding domain where Casirivimab and Imdevimab bind. Laboratory studies and then real world data indicated that the effectiveness of these REGEN-COV, like many other early monoclonal antibody therapies, was substantially reduced against Omicron and its sublineages. Now, the very site the antibodies were designed to target had changed significantly enough the antibodies could no longer bind and neutralize the virus as effectively. Consequently, the authorization was revised and its use became limited in regions where Omicron was a dominant circulating strain.
It truly highlights the dynamic nature of this battle. So, what are the key lessons we have learned from the experience with REGEN-COV and the antibody cocktail approach in general?
There are several important takeaways. Firstly, the cocktail strategy is a valid and powerful approach to potentially delay or prevent the emergence of drug resistance. For a period, REGEN-COV demonstrated this benefit. It showed that hitting the virus at multiple points can be more effective than a single line of action. And secondly, it underscored the sheer speed and the adaptability of the viral evolution. Even a dual antibody cocktail targeting distinct epitopes can eventually be overcome if the virus undergoes extensive mutation in those target regions, as we saw with the Omicron. This experience has really emphasized the need for continuous surveillance of new viral variants and rapid assessment of how existing therapies hold up. It also drives home the importance of developing next generation antibodies and antibody cocktails that target even more highly conserved regions of the virus. The part that the virus absolutely cannot change without losing its ability to function. We also need to think about the platforms that allow for even faster modification or development of new antibodies as major variants emerge.
It seems the lessons from REGEN-COV are shaping how we approach future antibody development, not just for COVID-19, but for other viral threats too. Before we conclude, a reminder to our listeners, our discussion is for informational purposes only and does not constitute medical advice. Please consult a healthcare professional for any health concerns. Dr. Aishwarya, this has been another incredibly informative session. Thank you for shedding light on the science behind REGEN-COV.
You're very welcome, Dr. Shikha. It's crucial to understand these scientific journeys.
And thank you to our audience for joining us on Neutralizing COVID, the monoclonal antibody story, and staying curious with us. If you enjoyed this conversation, don't forget to hit that subscribe button and share this episode with your colleagues and friends. Until next time, take care and keep exploring science.